to all of you for joining in here. As we all know that Indian government has exempted gene editing, especially SDN1 and SDN2 from Rules 7 to 11 of the Indian Environment Protection Act, thus giving the exemption from the applicants from the purview of getting approval from GEAC, Genetically Engineering Appraisal Committee. So this gives a bypass route from not getting approval for any gene edited crops under the existing GMO rules of India. So this is a cause of alarming. So for that, to build our understanding in the new technologies in gene editing process, we are making this a Skillshare capacity building workshops. So we have Dr. Michael and Dr. Raman JLU kindly agreed to help us improve our understanding in these two key interesting topics. हमारे भारत सरकार ने जीन एडिटिंग को स्पेशली SDN1 और SDN2 के रूल्स को एनवायरमेंट प्रोटेक्शन एक्ट के तहत एग्जेम्प्ट कर दिया है। इसका मायने एक जो भी नया एप्लीकेंट है जो जीन एडिटिंग क्रॉप्स या ऑर्गेनिजम्स को एनवायरमेंट में लॉन्च करना चाहते हैं, वो एग्जिस्टिंग GM रूल्स के तहत नहीं हो सकता, एग्जम्पशन मिल गया है, जिसका मतलब है वो GEAC के अंदर रूल्स को फॉलो करने की जरूरत नहीं है। तो इस विषय को ज्यादा समझने के लिए हमारे साथ डॉक्टर माइकल और डॉक्टर रामन जेलू हैं। The session flow, we will have Dr. Michael first and Dr. Raman Jelu. Dr. Michael will help us understand a brief about genetic engineering as a concept and some history and what is happening all over the world and what can we do as a collective. And Dr. Raman Jelu will take the topic of the brief history in India and what's the regulation is happening in India, and after this we can have a collective discussion and the steps ahead we can strategize हम as a community क्या कर सकते हैं। I will take this opportunity to introduce Dr. Michael. Dr. Michael is a leading genetist from London. He's from King's College London, and he is the head of Gene Expression and Therapy Group. And his research, he has been constantly researching on the topics of how gene editing is, today we are using the term that gene editing is very precise. So he is, through his research and talks, he is debunking the myths. So I'll not take more of time. Dr. Michael, over to you.
Thank you very much, Ajay. Namaskar to everyone, and I must first congratulate the organizers for taking the initiative to hold this very important webinar, and I thank the organizers for inviting me to speak. Just to give us a little bit more of my background so you can see where I'm coming from and why I was invited. I'm a career-long molecular geneticist studying gene structure and function and making many contributions in the field of biotechnology. So I'm familiar firsthand with all forms of genetic manipulation technologies, including more recently gene editing, but all within a clinical research application context. But it's from that background that makes me very concerned about how this technology has been applied and is planning to be applied, including the technology of gene editing within an agricultural context. So with this, I will now share screen of my presentation, and I hope you'll all be able to see it. Good, everybody good?
Yeah.
Good. So, title of my presentation, I will just get my pointer so that The title of my presentation is New GMOs, New Genetically Modified Organisms, Threats to Health and the Environment. First, let's go back to the beginning and define what is GM, genetic modification. Genetic modification, this is the European Union law definition, but really this definition has been basically adopted worldwide by different regulators. And that GM is a process where genetic material has been altered in a way that does not occur naturally by mating or natural recombination. Yes, the emphasis here is on does not occur naturally, this process. So this is why international protocols and treaties around the world recognize GM as different from conventional breeding and poses therefore different risks. And that therefore there are safety considerations to health and the environment that need to be taken into account because of the novelty of GM compared to natural breeding, which has been going on forever. So what is genetic engineering? Genetic engineering began in the 1970s. I remember when I was an undergraduate at Oxford University when it all began in the 1970s. And the first GM plants were introduced in the 1990s, first tomato, then soybeans, and then maize, cotton, and so forth. But what is important to understand right from the beginning is that genetic engineering is a laboratory procedure. So that involves direct intervention in the genetic sequence of the organism. So it's not a natural breeding process. It is an artificial laboratory procedure, as I will illustrate to you shortly. And that older style genetic engineering, just to put things into a historical context, is where you have the insertion of a foreign gene, foreign genetic material into the organism, which is then selected. And but the insertion and this insertion process does not generally occur naturally. Yeah, so and this now illustrates to you the process of genetically modifying a plant. As I mentioned, it's a laboratory-based procedure. So in step number one, the cells from the plant that you want to genetically modify are grown in dishes in the laboratory. This is a process here called protoplast tissue culture, plant cell tissue culture. Then you introduce into the plant cells growing on your dishes your DNA that you wish to insert into the plant cell DNA. And you use an artificial procedures called gene gun or biolistics or bacterial infection to introduce the foreign DNA, the plasmid. And then in step three, on rare occasions, this foreign DNA is inserted into the DNA of the plants. And then once you select for these rare events where the DNA has inserted, a whole plant can be grown from the transformed cells, the genetically modified plant cells, and so you end up with your mutant plant. And you screen then the plants for viability and for your genetic modification. And this should be very obvious to everybody that this is not a natural process. Hence, the need to assess the risks, because this bears no resemblance to normal breeding practices. So GM is radically different from conventional breeding. It allows, as you can see, because of the artificial nature of this process, it now allows the introduction of foreign genetic material, foreign genes from any organism into the plant, from different species, different families, and even different kingdoms. You could introduce human genes into a plant if you wanted. It is not a problem. And therefore, the GM allows the ability to create genetic material and products of that material that have never existed before. And therefore, radically differs from traditional breeding. But the key thing here is that GM, genetic modification, is highly prone to unexpected outcomes because it brings about new combinations of gene functions and disrupts the normal host gene function. Just to graphically illustrate to you here what can happen, this is a study conducted by Indian scientists where they basically crossed a variety of rice was produced that produces vitamin A, beta-carotene vitamin A in the grain, in the rice grains. So they are yellow actually. So they took this vitamin A rice and they crossed it into a local popular Indian variety of rice in India called Swarna. And what you can see here is that in the normal variety in Swarna, and when there was just the one chromosome copy of the GM genes, things looked normal. But when the pollination, when the crossing was undertaken in such a way that you now had two chromosome copies of the vitamin A rice genes, a state known as homozygous state, the rice grew very poorly and the yields were very low indeed, as you can see on the photographs of the rice heads on the right, and the stunted growth of the rice plants that you can see on the right. So this comes from unexpected genetic damage in the rice. So you can get from GM, you can get compromised crop performance, as well as I will show you later, unintended biochemical changes that can lead to toxicity and allergenicity. So I will stop there for, this lays a bit of historical context. Before I now go into the gene editing, maybe now we can have a brief translation for the non-English speakers. Can we have this, Ajay?
Yeah, Ishtiyaq ji.
Yeah, तो डॉक्टर माइकल ने अभी तक जो बातें कही उनको हम बहुत जल्दी में बता देंगे। जैसे जेनेटिकली मटेरियल जो है वो इस तरह से जेनेटिक मटेरियल को जीएम टेक्निक के जरिए इस तरह से बदल दिया जाता है कि जो प्रकृति में पहले से मौजूद नहीं है उसमें बिल्कुल नए किस्म की चीजें पैदा होती हैं। जेनेटिक इंजीनियरिंग की शुरुआत 1970 में हुई और पहला जीएम पौधा जो है वो 1990 में प्रस्तुत किया गया, यानी इंट्रोड्यूस किया गया। और ये इस तकनीक के तहत जो भी चीजें होती हैं वो अप्राकृतिक हैं और लैब में की जाने वाली प्रक्रिया है जेनेटिक मॉडिफिकेशन। इसके 4 चरण हैं जिसमें पहले इसमें चरण में टिश्यू कल्चर को लिया जाता है, उसमें फिर जीन गन के इस्तेमाल से बाहरी डीएनए को किसी दूसरे डीएनए को इस पौधे में के टिश्यू में इंसर्ट किया जाता है, यानी डाल दिया जाता है। इसके बाद से जो पौधा तैयार होता है वो बिल्कुल नए किस्म का होता है और उसका चरित्र बिल्कुल से अलग होता है। अगली बात जो महत्वपूर्ण बात उन्होंने बताई वो ये है कि जीएम से कई तरह के अनपेक्षित नतीजे भी निकल सकते हैं, है ना, कि कोई जरूरी नहीं है कि आप जो पहले से सोच करके आपने जो चीजें की हैं वही होगा हमेशा। उसमें बहुत सारी चीजें ऐसी भी हो सकती हैं जिसके बारे में आपने नहीं सोचा था और उस तरह के परिवर्तन आ सकते हैं। और उन्होंने काफी विस्तार से धान के मामले में बताया कि किस तरह से स्वर्णा धान के साथ जब ये किया गया तो उसमें कई तरह की समस्याएं देखने को मिली। थैंक्स।
Thank you very much, Ishte. So the question, now is new GM, including gene editing. Is it a step forward? A new generation of GM plants and livestock animals have been developed using this new GM techniques, which are sometimes called GM 2.0. And the most commonly used technique in gene editing these days is the CRISPR-Cas tool, which I'm sure you have all heard, because hardly a day goes by with some new claim as to what CRISPR-Cas gene editing will produce for the world. And those promoting these new techniques, especially gene editing techniques, often call gene editing precision breeding techniques or new plant breeding techniques. So this term precision breeding is something that I will challenge in coming presentation. The whole idea, the advocates of using gene editing in producing new plants and farm animals, the reason why they're trying to use terms like precision breeding or new plant breeding techniques is to actually avoid the stigma of the term GM or genetic modification, which is highly mistrusted in Europe and I believe elsewhere in the world. So it's really a semantics issue here, not a technical point that we're really dealing with here. But there's been, the reason, and this is the reason which brings us all here today, there has been a very powerful global lobby pushing to deregulate gene-edited products. This massive global lobby has been pushing to exclude and therefore de facto deregulate certain gene-edited products that come under the headings SDN1 and SDN2. And as was said earlier by Ajay, these outcomes of SDN1 and SDN2 gene editing has already been deregulated. In effect, they've been excluded from the current GMO regulations by the regulators in India. And this is also the case in other countries such as Australia, Japan, United States, and the UK is pushing for this right now as well. The UK government is trying to rush through legislation that will not only exclude SDN1 and SDN2 from GMO regulations, but actually all GM, which is really incomprehensible as it is so unscientific. The principal argument, the advocates for deregulation of gene editing, the principal arguments they use are as follows. They argue that small DNA base unit changes, SDN1 and SDN2 changes, mimic what may occur naturally through random mutation. That the intended changes in the genes are precise. Only what you design happens, and that no other gene alterations occur in the target organism. No foreign DNA is inserted into the plant or the animal, and that the outcomes are totally predictable, and thus the products are completely safe. And the only way, I should add, that they can make these types of assertions is this underpinning tenet, which is they will focus your attention exclusively on the end product and ignore the process by which the gene editing is undertaken. So in other words, you make the change and they say, just focus on this change I've made. Don't worry how. Don't concern yourself by how I made the change. Just focus on the change itself. Now this, as I will, in other words, end product and not process focus. This I will lead you through. Hope you'll find is just about the most unscientific position you can take. So the question is, are these claims supported by the scientific evidence that underpins this technology? So first, what is gene editing? Gene editing is a genetic modification technique that is designed to make a targeted alteration in the DNA of an organism, whether it be plant or animal. Most often, gene editing involves cutting the DNA across the two strands, what is known as a double-strand DNA break. And this double-strand DNA break is the gene editing is designed to bring this about at a predetermined location using the gene editing tool. And so the outcome, there are three possible outcomes from the gene editing tool application. First, gene disruption, where you literally cut the gene and there's imperfect repair and you destroy the gene function. Gene disruption, or destroying a gene function. Secondly, there's gene alteration. So you change the information in the gene so that now it produces a new protein product. And lastly, you can use gene editing to insert a whole new gene as well at a predetermined location. So how does gene editing work? There are two approaches. The one that we will focus today is the use of what are known as site-directed nucleases, or SDN. This is where the terms SDN1, SDN2, SDN3 derive from. So site-directed nucleases, there are four main types. The one that have really, there is meganucleases, zinc finger nucleases, and Taylor nucleases. But the one that has captured the imagination of the world is the CRISPR-Cas system. Now, fundamentally, all of these different nucleases, all of these different SDNs work in exactly the same way. So what you do is you take your cells from your organism, plant, animal, and you treat them with your nuclease, your SDN. It could be zinc finger nuclease, TALEN, or the CRISPR-Cas SDN. And these produce, as I say, in most applications, not all, but in most applications, what this is designed to do is to cut the DNA, produce a double strand break, a DSB here. And now after this is where the precision, at least the degree of precision, goes. What I'm about to tell you from now on happens beyond the complete control of the gene editor, which is where the dimension of imprecision now comes in. So in SDN1, after the double strand cut by the SDN, we have the repair of the break. The cell tries to repair the break, but in trying to rejoin the DNA, broken DNA strand, it is imprecise. And so you end up with either a loss of some base units of DNA or the insertion of some base units of DNA, deletion or insertion. And this is what then results in the destruction of the normal information content of the gene and destroys the production of the normal protein. In SDN2, the repair of the cut DNA is aided by the introduction of a second DNA molecule, what's called the repair DNA. The repair DNA carries the same sequences as where the double strand cut has taken place. And this encourages a process of DNA repair known as homology-directed repair. Homology-directed repair, as illustrated in the central part of this diagram, is where you get exchange of the DNA of the host DNA with the DNA you've introduced into the plant cells, the repair DNA. So now you can introduce an altered sequence through the repair DNA exchanging with the host DNA. You can introduce a genetic modification in the gene. And that's SDN2. And SDN3 is where the repair DNA carries a whole new gene. So now you introduce a whole new gene. And as mentioned earlier, it is the SDN1 and the SDN2 which have been deregulated in India and have been deregulated in other nations around the world. And if the UK government gets its way, it will be deregulated in the UK as well. And in terms of how you conduct gene editing, I'm not going to dwell very much on this slide because you've seen it before. The whole procedure of gene editing is an artificial laboratory procedure. Exactly the same steps are involved, where you grow the plant cells, introduce DNA that codes for
Dr. Michael, should we go for a translation for first this part?
Can I finish this slide?
Okay.
And then we take a break for the translation, because then there's a natural break. So you introduce, just like when the old style GM, where you introduce foreign gene, here you introduce into your plant cells DNA that codes for your CRISPR-Cas gene editing tool, which then carries out the gene editing, and then you grow the mutant plants from here. Again, you can see that it is not a natural breeding procedure, but a total artificial genetic manipulation procedure. So now we can take a break for some translation.
Thanks, Dr. Michael. डॉक्टर माइकल ने एक बात बहुत साफ तौर पे कही कि जीन एडिटिंग, हालांकि इसको एक नया नाम जरूर दिया गया है, जीएम की ही एक नई तकनीक, जेनेटिक मॉडिफिकेशन की ही एक नई तकनीक है, जिसमें क्रिस्पर कैस टूल का इस्तेमाल होता है। इसमें प्रिसीजन ब्रीडिंग टेक्निक का इस्तेमाल होता है ना, और इसमें फर्क सिर्फ इतना है कि यहाँ पे हम कोई नया डीएनए, किसी और जीव का डीएनए इसमें नहीं डालते हैं, बल्कि उसी प्लांट के डीएनए में या वो फेर बदल करते हैं, और बस इतना ही फर्क है। दरअसल ये एक तरह का जेनेटिक मॉडिफिकेशन ही है, लेकिन दुनिया में चूंकि जेनेटिक मॉडिफिकेशन को लेकर के एक काफी ज्यादा अविश्वास है और उसको लोग पसंद नहीं कर रहे हैं, तो उसको जस्ट एक नाम बदल करके एक नए ढंग से इसको पेश किया जा रहा है, जिसमें ये कहा जा रहा है कि हम डीएनए को के साथ, यानी डीएनए हम नया नहीं डाल रहे हैं, इसलिए आपको परेशान होने की जरूरत नहीं है। इंडिया में जैसे एसडीएन और एसडीएन1 और एसडीएन2 को अभी तक इजाजत मिली है, तो डीरेगुलेट किया गया है। दुनिया में कई जगहों पे इन्हीं सारे तर्कों के कारण डीरेगुलेट किया गया है। इसमें भी फिर डॉक्टर माइकल ने पुराने स्लाइड को जो उन्होंने जेनेटिक मॉडिफिकेशन के तरीके के लिए जो स्लाइड यूज किया था, उसको दिखाते हुए ये बताया कि इसमें तरीके सिर्फ अलग अलग हैं। जैसे एसडीएन1 में करते हैं क्या कि वो कट करते हैं एक जीन को, और उसमें वहां पे रिपेयर किए बगैर उसको वहां कट करके उसको जोड़ने की कोशिश करते हैं। और एसडीएन2 में क्या है कि वो डीएनए को रिपेयर भी करते हैं और फिर उसको म्यूटेट करते हैं। एसडीएन3 में है कि सीधे वो एक सेट को सीधे रिप्लेस कर देते हैं। तो अभी तक जो इंडिया और कई दूसरे देशों में इजाजत मिली है, वो एसडीएन1 और एसडीएन2 को मिली है। अभी इंग्लैंड में भी उसकी कोशिश की जा रही है और वो प्रक्रिया शायद चल रही है। That's it, I guess.
इश्तियाक, एक इम्पोर्टेंट बात जो आपने कहा जाता है कि फॉरेन जीन इसमें नहीं आएगा, और डॉक्टर एंटोनियो ने ये बताया कि इसमें भी वो फॉरेन जीन आने की संभावनाएं काफ़ी है और वो बताने वाले हैं। और एसडीएन2 से लेके उन्होंने दिखाया कि वो दूसरा जीन्स भी आ सकते हैं।
बिल्कुल, और उन्होंने जो सबसे महत्वपूर्ण बात बताई कि इससे भी यानी जीन एडिटिंग से भी एक अप्राकृतिक प्रोडक्ट ही पैदा होता है, वो अननेचुरल जैसे कि जेनेटिक मॉडिफिकेशन में होता है, वो भी जीन एडिटिंग से भी वैसा ही अननेचुरल या अप्राकृतिक चीजें ही निकल करके आती हैं नतीजे के तौर।
वो जहां काटना चाहते हैं वो काटेंगे, मगर बाकी जो डिलीशंस होते हैं और इंसर्शंस भी होते हैं उसके बारे में वो बात नहीं करते हैं, मगर वो प्रक्रियाएं भी होती है। और इस ढंग से देखा जाए तो ये भी बहुत अप्राकृतिक हो रहा है।
थैंक्स कविता।
Dr. Michael,
Yes.
over to you.
Shall I continue?
Yes, over to you.
Okay. So with this background on the basics of gene editing, what is gene editing and what is involved and how it is undertaken, let us move forward. Some examples of gene edited crops that you may have heard of is the American company Calyx in the United States has commercialized a variety of soybean with altered fat composition and is developing a low gluten wheat. Corteva, a major biotech company that actually holds the patent rights to gene editing use in agriculture, they have altered maize to change the starch composition, and this has been commercialized in a number of different nations. A scientist at Penn State University in the United States has used the CRISPR gene editing tool to produce mushrooms that do not brown when you cut them. In Japan, there has been commercialized recently a variety of tomato that produces GABA, which has a sedative type effect. Not sure how useful that is, any more than non-browning tomatoes, if you ask me. And in England, there's been a lot of publicity recently of a tomato produced by the John Innes Center, which produces vitamin D. Although this has been rather, in my view, deceptively described, because it's not vitamin D that gets produced in the tomato, but a chemical that can be converted to vitamin D. But anyway, this is just some examples. The wish list of what gene editors can say they can produce is as long as your arm. But the point is, it's all a wish list. Little has actually been proven to be possible with this technology. In terms of gene edited farm animals, you may have heard of the hornless cows, where although there are naturally occurring hornless cows, so I don't know why they even bothered to produce this. And then researchers in South Korea have destroyed a gene function in pigs that normally regulates muscle growth. So now these poor pigs grow grotesquely large muscles, which poses major animal welfare problems. These are just a few examples. The first, however, just to begin to address the issues of the technology of gene editing, there's first of all the claim of precision. This is the very first claim of gene editors in agricultural context, that everything is precise, targeted, and predictable. But actually, gene editing is not precise. Gene editors claim that gene editing introduced changes are similar to what may occur naturally, but this is unproven and is an untested hypothesis. So claims of naturalness are highly questionable. What is known about gene editing, it is not precise in that the gene editing tool, especially the CRISPR-Cas tool, can produce DNA damage, mutation, unintended, unpredictable DNA damage, both at off-target and on-target sites. What do I mean by this? Off-target DNA damage is locations in the DNA of the organism that you do not intend to take place. And on target is the intended gene edit site. So you can have unintended DNA damage where you are trying to edit and where you are not trying to edit. This will happen invariably. Off-target means damage occurs at locations of the genome other than the intended gene editing site, and on-target means unintended damage at the intended edit site. And the overall message, however, mutations are a bad thing, yeah? You may, the advocates of gene editing, which are intentionally trying to cause mutations in plants and animals, is that somehow mutations are natural, they're normal, and to be welcomed because they form part of natural evolutionary processes. This is simply unscientific, not true. Mutations are recognized by regulatory agencies around the world to be not good, which is why we have laws in place everywhere to protect us against DNA damaging agents such as radiation and chemicals that can cause DNA damage. So mutations are to be avoided, not to be intentionally made in plants and animals or even humans. So currently recognized gene editing unintended mutational effects. So gene editing can bring about unintended DNA damage, as I mentioned, at off-target mutational effects. In other words, DNA damage at locations where you do not wish any damage. And this DNA damage, of this off-target DNA damage mutations will not be random, but in other genes when you use these gene editing tools, especially the CRISPR-Cas tool. And the recognized on-target mutations are numerous. I don't have time, but obviously I have references with me that will back up every one of these statements that I'm making here. You have unintended side effects from the intended alteration. You change a protein function, an enzyme function, and it can carry out chemical reactions other than those that you are intended, and this can have major consequences on the organism and the consumer. Large DNA deletions affecting more than one gene, large DNA rearrangements affecting multiple gene functions, creation of new gene sequences resulting in new RNA and proteins and mutant proteins, insertion of plasmid DNA encoding the gene editing tool or contaminating DNA, even when the foreign DNA is not intended to be inserted. So this idea that SDN1 and SDN2 does not introduce foreign DNA, that's the way you design it. But very frequently, that will not be the case, and foreign DNA will be inadvertently inserted. And all of these types of mutations here that I've listed can occur at both the off-target sites as well as the on-target sites. So what we have is at all, in this slide, I'm summarizing where the unintended, unpredictable, unintended DNA damage mutations can take place during the gene editing procedure. It takes place at the stage of the simple plant tissue culture, where you're just growing the tissue culture cells. They take place when you introduce the gene editing tool into the plant cells. And then they take place from the gene editing tool itself as well. The gene editing tool itself has unintended off-target and on-target mutations. So at each, so the point I want you to bear in mind here, at each of the steps and components of the gene editing process, the process here, yeah, you get unintended DNA damage. So it accumulates. You get an accumulation of unintended DNA damage at all the different steps of the gene editing process. And I will finish this slide and then we can have a translation. And that the on-target mutations are not controllable. Why is this? It's because the on-target mutations occur after the gene editing tool has finished its task. The gene editing tool cuts the DNA and it's finished with its task. What happens after that is beyond the control of the gene editor, because one is at the mercy of the cell's own DNA repair mechanisms and machinery. So no matter how precise you make that initial cut of DNA with the gene editing tool, the unintended on-target mutations can still take place. So this, I will stop for translation at this point.
थैंक्स डॉक्टर माइकल। डॉक्टर माइकल ने कई एग्जाम्पल्स बताए जीन एडिटिंग के, जिसमें उन्होंने बताया कि कैलिक्स नाम की एक कंपनी ने यूएस में ऑल्टर्ड फैट सोयाबीन, जो अभी कमर्शियलाइज भी हो गया है, यानी खुले बाजार में, यानी उसका बीज मिल सकता है और लोग उगा रहे हैं, उसका व्यापार शुरू हो गया है। दूसरी चीज जो अभी अंडर डेवलपमेंट है वो लो ग्लूटेन व्हीट को वो कैलिक्स नाम की कंपनी तैयार करने में लगी हुई है। इसी तरह से कॉर्टेवा जो पहले ट्यूपॉन्ट के नाम से जाना जाता था, वो भी अमेरिका में ही है। वो क्रिस्पर जीन एडिटिंग के जरिए वैक्सीमेज को तैयार करने की कोशिश कर रहा है। इसी तरह से पेन स्टेट की जो अपनी यूनिवर्सिटी है, यानी उस राज्य की जो अपनी यूनिवर्सिटी है, ये भी यूएस में ही है, अमेरिका में ही है। वो भी जीन एडिटिंग के जरिए ऐसा मशरूम पैदा करने की कोशिश कर रहे हैं जो ब्राउन नहीं होता है, यानी भूरापन जिस पे नहीं आता है, वो हमेशा सफेद दिखता है। इसी तरह से जापान में गाबा टोमेटोज पैदा कर रहे हैं, जिसमें सिडेटिव की मात्रा काफी हाई होगी। सिडेटिव मतलब जिससे आप यानी नींद में मदद कर सकता है वो, या आपको शांत करने में वो मदद कर सकता है। इसी तरह से यूके की एक कंपनी है जॉन इंस सेंटर, कंपनी नहीं है बल्कि सेंटर है, रिसर्च सेंटर है। वहाँ पे टोमेटो में विटामिन डी की मात्रा बढ़ाने वाली तकनीक इस्तेमाल की जा रही है। इसी तरह से बिना सींग वाली गायों को पैदा करने की कोशिश, यानी पैदा किया गया है, जबकि डॉक्टर माइकल का कहना है कि बिना सींग वाली गायें पहले भी मिलती हैं, यानी प्राकृतिक ढंग से मिलती हैं, तो इस पे इतनी ज्यादा मेहनत करने की जरूरत क्या है। इसी तरह से नीचे में आप देख रहे हैं कि सूअर हैं जिनके शरीर में बहुत अलग ढंग से यानी अप्राकृतिक ढंग से मसल्स, यानी मांसपेशियां उसकी विकसित की गई हैं। इस तरह की छेड़खानी के जरिए ऐसे कुछ नए जीव, यानी जो पहले से यानी एक नए तरह के जीव पैदा करने की कोशिश है। इसमें डॉक्टर माइकल बहुत साफ तौर पे बता रहे हैं कि ये, जिसके बारे में बार बार कहा जाता है कि ये बहुत प्रिसाइज है, और प्रिसाइज का मतलब है बहुत लक्षित ढंग से आप इसको कर सकते हैं। जो नतीजे आप चाहते हैं वही नतीजे आप हासिल कर सकते हैं। ये कहा जाता है, ये दावा है, लेकिन सच्चाई ये है कि ऐसा हो नहीं रहा है। ये इसका अभी तक कोई वैज्ञानिक प्रमाण नहीं है, और ये अभी भी एकदम पूरी तरह से हाइपोथेसिस के लेवल पे ही है। और इसमें दो तरह के टारगेट्स होते हैं, ऑफ टारगेट होता है और ऑन टारगेट होता है ना। और ऑफ टारगेट का मतलब है कि जहाँ पे जो डैमेज है वो लोकेशन पे जीनोम के अंदर डैमेज हो सकता है, और जो कि पहले से आप चाहते नहीं है, अनइंटेंडेड जो है ना। ऑन टारगेट का मतलब है कि जो आप चाहते हैं, है ना, वैसा ही चेंज होता है। उनका कहना है कि म्यूटेशन अपने आप में एक खराब चीज है, उसको अवॉइड करना चाहिए, उसको प्रमोट नहीं करना चाहिए। इसी तरह से वो आगे बता रहे हैं कि बहुत सारे केसेस में ये जो अनइंटेंडेड यानी जो गैर इरादतन जो बदलाव आएगा, यानी जो म्यूटेशन हुए हैं, है ना, वो टारगेट जीन्स में भी देखे गए, जिसमें प्लांट टिश्यू कल्चर में ही म्यूटेशन देख सकते हैं। और ये बता रहे हैं कि ये चारों जो स्तर हैं, जो पहले जेनेटिक मॉडिफिकेशन के भी जो 4 स्तर हैं, ये अनइंटेंडेड जो म्यूटेशन है वो चारों में से किसी भी लेवल पे हो सकते हैं। इसलिए ये जो बार बार कहा जाता है कि ये एकदम प्रिसाइज है, इसमें किसी तरह की गलती की कोई गुंजाइश नहीं है, वो एकदम से गलत बात है। और नतीजे आम तौर पे या उसमें पूरी आशंका इस बात की है कि नतीजे बिल्कुल जो आप चाहते हैं उसके खिलाफ जाए। और ये यहीं पे रुकता नहीं है। और इसमें एक बात जो उन्होंने कही है कि जेनेटिक जब एडिटिंग कर रहे हैं उस वक्त आप जितना भी प्रिसाइज होने का दावा कर लीजिए, लेकिन एक बार जब वो प्रक्रिया पूरी हो जाती है तो उसके बाद एडिटर का कोई कंट्रोल उस पर नहीं रह जाता है, और उसके बाद किसी भी तरह का उसमें बदलाव आ सकता है, किसी भी तरह का अनइंटेंडेड जो बदलाव हैं, वो अनअपेक्षित या गैर लक्षित जो बदलाव हैं, उनकी पूरी आशंका मौजूद रहती है। इसलिए आप कितना भी कहिए कि डीएनए कट कितना भी प्रिसाइज हो, लेकिन वो उसमें बहुत सारे ऐसी गुंजाइश रहती है जिसमें जो आप नहीं चाहते हैं वो भी हो सकता है। That's it.
Yeah. Thank you very much. Thank you very much, Ishta. So, unintended outcomes from the intended gene edit. Yeah. So even from changing a single gene, you can have unintended outcomes, because no gene works in isolation. It works as part of a whole genome network. So altering the function of just one gene can have large effects on the genomic network and, of course, the products that the genomic gene network produces. And this, as a result, one can end up with large-scale changes in the patterns of gene function, which can radically alter chemical composition and functioning of the plant in totally unpredictable ways. GMO gene editing developers largely ignore this possibility because they fail to undertake the in-depth molecular compositional and functional profiling, compositional analysis that is needed to address these risks. So this is a major regulatory developer and regulatory failing. The risks of gene editing vary from case to case. So you may wonder, so we get all this DNA, unintended DNA damage. So why should we be worried? What is the reasons as to why we should be worried? The unintended mutations, both on target and off target gene editing sites, can lead to altered patterns in gene function, which in turn can lead to unintended changes in the biochemistry of the organism. Okay, because patterns of gene function is what controls the biochemistry and composition of the organism. So if the patterns of gene function change, there will inevitably be changes in biochemistry and composition. And in plants, in gene-edited plants, these unintended changes in gene function can lead and composition can lead to the production of unexpected toxins and allergens or compromised nutritional value. So here is the health implications of this unintended DNA damage. And that Now, you may ask, is there any evidence that gene-edited foods have been found to have health, result in adverse health? And the answer is, we don't know, because nobody has actually conducted the research, the toxicity studies, to evaluate the composition and safety of gene-edited plants through toxicity studies, at least say in animals. So it's all the claims of safety of gene-edited plants is completely hypothetical. It's an assumption. It's not evidence-based. What we do know is that there are many animal feeding studies of first-generation GM crops, transgenic GM crops, that showed toxic and allergic type effects. And given the overlap in the two technologies, the processes of the two technologies, old style transgenic and new style gene editing, and the kinds of alterations in unintended gene function they can produce, it is not beyond the realms of possibility that one will find toxic and allergic effects from gene-edited products, just like one found from first-generation transgenic GM crops. We have to remember, for example, that plants are very good at producing toxins. That plants naturally produce toxins to protect themselves from predators. So changing the biochemistry of a plant through unintended or even intended mutations, it is not beyond the realms of possibility that that plant will now start producing a new toxin or high levels of a known toxin. And also, remember another claim of the advocates for deregulating SDN1 and SDN2 is that no foreign DNA is inserted into the animal or the plant through SDN1 and SDN2. But actually, this can happen inadvertently, very frequently. So for example, in the hornless cattle, hornless cows, gene-edited hornless cows I mentioned earlier, which actually is an SDN3 outcome, there was inadvertent incorporation of foreign DNA, the plasmid DNA that was used to drive this SDN3 process. The whole plasmid DNA was inserted, which meant that antibiotic resistance genes were incorporated into these animals. And also this can occur in plants. This study from 2016 by 2 Korean scientists, bypassing GMO regulations with CRISPR gene editing. What they found was that there was frequent insertion of gene editing encoding plasmid fragments in the plants. And these are not being looked for. My point here is that these are not being looked for. And so you can end up marketing a gene-edited product which you claim to be SDN1 and SDN2 with no foreign DNA, but actually there is inadvertent insertion of foreign DNA there. It's just that because the regulator didn't ask you to look for it, you didn't look for it, and it's there and could have health implications. So gene editing is basically a genetic modification procedure. And so when the European Court of Justice ruled in 2018 that gene editing, which it called new mutagenesis techniques, was indeed GM and gives rise to genetically modified organisms, GMOs, and therefore fall under the scope of the EU GMO regulations, the European Court of Justice was being completely true to the science that underpins gene editing technology. And that the risks from the use of gene editing could be similar, if not greater, than older style GMOs. So to bring this all together, given that gene editing is a laboratory-based artificial genetic modification procedure, does not in itself involve natural crossbreeding, results in functional alterations of one or more DNA sequences, causes unintended and unpredictable off-target and on-target effects of DNA, RNA, and protein levels. Gene editing is a GM procedure, and regulations applied to their products should be process-based as well as product-based, as is currently the case in the EU and currently in the UK, but as I say, the UK government is trying to change that. And that and why do we need process-based regulation? Basically, briefly, process informs you when the process of how you bring about a genetic modification informs you about not only how you bring about what you want, the intended genetic change, but the process informs you of unintended alterations of DNA damage. And therefore it informs regulators, process better informs regulators to enact rules that would better protect the public and the environment. And process-based regulation is true to the state of the science and technology. And any attempts to argue that such process, as well as product-based regulation, are superfluous or excessive, is being disingenuous and puts public health and environment at risk. And so I'm afraid the Indian government, by deregulating SDN1 and 2, this is exactly what it's been. It has been disingenuous, it has been dishonest and disingenuous to the science that underpins gene editing and is putting public health and the environment at risk. I'll stop here for a translation.
Thanks, Dr. Michael. डॉक्टर माइकल ने इस बात पे काफी जोर दिया कि जीन्स जो होते हैं वो अकेलेपन में, आइसोलेशन में कभी काम नहीं करते हैं, वो एक नेटवर्क के तहत काम करते हैं। इसलिए किसी एक जीन को भी अगर आप उसके साथ कुछ हेर फेर करते हैं, उसमें बदलाव करते हैं, तो ये पूरे जीनोमिक नेटवर्क को इम्पैक्ट करता है। और इस तरह से वो कई बार पूरे के पूरे जो पूरा ऑर्गेनिज्म है या जो जीव है, चाहे वो पौधा हो चाहे वो जानवर हो, उसके पूरे जेनोमिक नेटवर्क को पर उसका असर होता है, जिसकी वजह से उसका पूरा केमिकल कंपोजिशन या जो रासायनिक प्रक्रियाएं हैं वो उन सबको वो बदल सकता है। और फिर आप ये तय नहीं कर सकते हैं कि वो किस तरह से बदलेगा, और वो पूरी तरह से अनप्रेडिक्टेबल होता है, जिसकी आप पहले से इसका अंदाजा आप नहीं लगा सकते हैं। तो इसी तरह से इसके खतरे जो हैं वो बहुत स्पष्ट रूप से सामने है कि ये पूरे बायोकेमिस्ट्री को बदल दे सकता है। किसी एक ऑर्गेनिज्म के पूरी बायोकेमिस्ट्री जब बदल जाती है तो फिर वो बायोकेमिस्ट्री किस दिशा में आगे बढ़ेगी उस पर कोई नियंत्रण जीन एडिट है। जीन एडिटेड जो पौधे हैं उनसे मिलने वाले भोजन में कई तरह के अनएक्सपेक्टेड, यानी जिसकी उम्मीद नहीं होती है, उस तरह के टॉक्सिन, जहरीले पदार्थ या एलर्जेंस पैदा हो सकते हैं, या वो खाने की जो न्यूट्रिशनल क्वालिटी है, उसकी जो पोषण क्षमता है उसको भी घटा सकता है। जो जानवर अभी तक फर्स्ट जनरेशन के जी क्रॉप्स खा रहे हैं उनमें कई तरह के टॉक्सिक और एलर्जिक टाइप के प्रभाव देखे जा रहे हैं अभी। इसमें उन्होंने ये भी बताया है कि टॉक्सिसिटी स्टडी चूंकि हुई नहीं है, इसलिए इसके बारे में कोई जानकारी भी नहीं है ना। उसके बाद जो ये दावा किया जाता है कि एक्सटर्नल डीएनए नहीं यूज कर रहे हैं जीन एडिटिंग में, यानी कोई बाहर से लिया हुआ डीएनए नहीं यूज कर रहे हैं, तो उन्होंने बताया कि ये दावा भी सही नहीं है, क्योंकि ये जो तस्वीर है जिसमें बिना सींग की जो गाय है, इसमें एक्सटर्नल डीएनए का इस्तेमाल हुआ है। इसलिए इसके तहत जो कई तरह के दावे हैं वो दावे अपने आप में गलत हैं। इसके आधार पे वो ये कह रहे हैं कि जीन एडिटिंग दरअसल जेनेटिक मॉडिफिकेशन ही है। 2018 में यूरोपियन कोर्ट ऑफ जस्टिस ने ये फैसला भी दिया है। अपनी फैसले में उन्होंने कहा है कि ये नए तरह का म्यूटाजेनेसिस टेक्निक है ना, और दरअसल ये जीएम है, और इससे जीएमओस को बढ़ावा मिलेगा, जो कि यूरोपियन यूनियन में जिस पर काफी सारा नियंत्रण है। तो हाँ, उन्होंने ये भी कहा है कि इससे वैसे ही बुरे नतीजे हो सकते हैं जो अभी तक पुराने जो जेनेटिक मॉडिफिकेशन प्लांट्स को खाने में जो खतरे थे वो इसमें भी हो सकते हैं। तो इसके बारे में वो कह रहे हैं कि ये भी पूरी तरह से लैब बेस्ड है, यानी लेबोरेटरी के अंदर में तैयार किया हुआ है। इसमें आर्टिफिशियल डीएनए मॉडिफिकेशन की प्रक्रियाएं इस्तेमाल होती हैं। इसमें प्राकृतिक क्रॉस ब्रीडिंग नहीं होती है, इसलिए कई तरह के फंक्शनल ऑल्टरेशंस इसमें संभव हैं, जो कि एक या कई डीएनए के सीक्वेंसेस में कई तरह के बदलाव ला सकते हैं। तो उनका कहना है कि दरअसल ये जीन एडिटिंग भी एक तरह की जेनेटिक मॉडिफिकेशन की प्रक्रिया ही है, और इसमें जो रेगुलेशंस हैं उनको उतनी ही सख्ती से, उतनी ही मजबूती से करने की जरूरत है, और इसको प्रोसेस बेस्ड यानी प्रक्रिया आधारित होना चाहिए, नतीजे पे आधारित नहीं होना चाहिए, जो कि बार बार जीन एडिटिंग के जो प्रणेता लोग हैं जो कहते हैं कि आप आम से मतलब रखिए ना, गुठलियों में क्यों जा रहे हैं। तो डॉक्टर माइकल का कहना है कि प्रक्रिया बहुत इम्पोर्टेंट है। प्रक्रिया के को जाने बगैर हम ढंग से उसका रेगुलेशन नहीं हो सकता है, और जो जेनेटिक मैनिपुलेशन की प्रक्रियाएं हैं उनको देखने का नजरिया या देखने का तरीका ही पूरा गलत हो जा सकता है। इसलिए उनका कहना है कि अगर रेगुलेशन की जरूरत है तो हमको प्रक्रिया आधारित रेगुलेशन होना चाहिए, सिर्फ नतीजे पर ध्यान नहीं देना चाहिए।
Thank you very much. Just in case you were wondering, I just got a couple more slides. I've almost finished. It's in case you're wondering, am I the only molecular geneticist that challenges or the position taken by the advocates of gene editing in agriculture? No, that's not the case. In some years ago now, the European Network of Scientists for Social and Environmental Responsibility published a statement challenging those advocating deregulation of gene-edited products. I'm a member of ENSA, and I was co-signatory of this. And more recently, I have led a sign-on statement for scientists and policymakers opposing this whole concept of precision breeding and why it is completely unscientific, and the term precision breeding should never be used to describe gene editing procedures. And I invite any scientists and those with policy background to read our statement and sign if they so wish. So just to summarize, bring everything together, your take-home messages from my talk: gene editing of crops and foods and animals is an artificial laboratory-based genetic modification procedure, and therefore by definition gives rise to genetically modified organisms. It produces unintended widespread DNA damage, off-target mutations, can result in unintended mutations at the intended gene editing site, on-target mutations, results in off-target and on-target mutations can combine to change the function of many genes, leading to altered biochemistry, including production of toxins and allergens. And therefore, for the above reasons, claims of naturalness, precision, and predictability for gene editing technology is not supported by experimental evidence. Gene editing cannot be called precision breeding. Claims of safety of gene edited foods is hypothetical because no safety testing has been conducted, although there is good evidence exists showing health risks from first generation GM foods. Gene editing of crops and foods and animals needs to be both process-based and product-based regulation to account for risks of unintended mutations. And that arguments for deregulation are not true to the science that underpins gene editing and put public safety and environment at risk. Therefore, we respectfully request that you oppose any attempt to exempt gene-edited organisms from GMO regulations and labeling. Thank you very much.
डॉक्टर माइकल ने ये भी बताया कि यूरोपियन यूनियन के 61 वैज्ञानिक इस बात से सहमत हैं कि ये जो नए किस्म के जीएमओज हैं उन पर पूरी सख्ती से उनका रेगुलेशन होना चाहिए और कई इस तरह के पिटिशंस साइंटिस्ट्स की ओर से और पॉलिसी मेकर्स की ओर से वहाँ शुरू किए गए हैं जिसमें वो कह रहे हैं कि ये प्रेसिजन ब्रीडिंग की जो बात है उसका वो विरोध कर रहे हैं। तो उन्होंने ये लिंक्स भी शेयर किए हैं, हम लोगों से साझा किए हैं ताकि हम लोग उनको पढ़ें और समझें और हम में से जो वैज्ञानिक हैं या जो पॉलिसी मेकर्स हैं उनको हम कन्विंस करें कि वो भी इन पिटिशंस पे, इन स्टेटमेंट्स पे अपना दस्तखत करें। फिर अंत में उन्होंने एक नटशेल में बातें कही हैं जो पूरे प्रेजेंटेशन को उन्होंने एक जगह पे बहुत संक्षेप में जो बातें कही हैं उनको एक जगह फिर से रखा है। वो कह रहे हैं कि जीन एडिटिंग भी आर्टिफिशियल है, लैबोरेटरी बेस्ड है, एक और जेनेटिक मॉडिफिकेशन की प्रक्रिया ही है। इसमें पूरी तरह से नए जेनेटिकली मॉडिफाइड ऑर्गेनिजम्स पैदा होते हैं। इनसे भी कई तरह के गैर लक्षित या अनपेक्षित नतीजे निकल सकते हैं और कई तरह के डीएनए डैमेज व्यापक स्तर पे देखे जा रहे हैं या उसकी पूरी आशंका बनी हुई है। ये ऑफ टारगेट में भी डीएनए डैमेज के केसेस हो सकते हैं और ऑन टारगेट म्यूटेशन के जो साइट्स हैं वहाँ भी कई तरह के अनअपेक्षित म्यूटेशंस देखे जा सकते हैं। तो इनका कहना है कि ऑफ टारगेट हो या ऑन टारगेट म्यूटेशंस दोनों ही हालत में पौधे की या जीव की जो बायोकेमिस्ट्री है उसमें चेंज की पूरी आशंका है, जिसके वजह से कई तरह के जहर और कई तरह के एलर्जेंस उसमें उनमें पैदा हो सकते हैं। नेक्स्ट स्लाइड या थैंक्स। तो इन सब को देखते हुए ये जो दावा किया जा रहा है कि ये कुदरती ढंग से होता है, इसमें बहुत सिद्ध ढंग से या बहुत प्रिसीजन के साथ हो सकता है ये काम, या फिर इसमें सब कुछ अपेक्षा के अनुरूप ही होता है, तो ये दावे अभी तक के जो प्रयोग हुए हैं उनके आधार पे कोई ऐसा साक्ष्य हमारे सामने नहीं है, कोई ऐसा एविडेंस हमारे पास नहीं है। इसके कारण ये कहते हैं कि जीन एडिटिंग को प्रेसिजन ब्रीडिंग इसीलिए नहीं कहा जा सकता है क्योंकि अभी तक उसमें बहुत सारी चीजें स्पष्ट नहीं है। और इसमें जो कहा जा रहा है कि जीन एडिटेड फूड जो है या भोजन जो है वो पूरी तरह से सुरक्षित है, इसका भी कोई वैज्ञानिक आधार अभी तक नहीं है क्योंकि अभी तक कोई सुरक्षा की टेस्टिंग ही नहीं की गई है। तो इसलिए ऐसा कहना बहुत ही सही नहीं है। और हाँ, इसका रेगुलेशन के बारे में वो कहते हैं कि इसको प्रोसेस बेस्ड और प्रोडक्ट बेस्ड रेगुलेशन होना चाहिए पूरी तरह से, ताकि उसमें कहीं कोई खामी रहे तो उसको समय रहते ठीक किया जा सके या उस पर रोक लगाई जा सके। तो इसलिए जो डीरेगुलेशन के लिए जो दावे पेश किए जा रहे हैं या जो आर्गुमेंट्स पेश किए जा रहे हैं वो साइंस के नज़र से वो अभी तक सही नहीं है। इसलिए उनकी ये अपेक्षा सबसे है, हम सबसे भी है कि जीएमओ रेगुलेशन और लेबलिंग को लेकर के काफ़ी सख्ती से उसको रेगुलेट किए जाने की ज़रूरत है। और जो डीरेगुलेशन की प्रक्रियाएं हैं उनको उनका विरोध किया जाना चाहिए। थैंक्स।
Thank you, Dr. Michael. Thank you. We have received few responses saying that we have not studied biology after school, but still we can follow. We were able to follow. So thank you very much, Dr. Michael. I have many questions, but I will just pick 3-4 questions before we move on to next segment. And the remaining questions we'll take it in the towards the end. Doctor, there's a question from Mr. Kapil Shah. How gene editing is different from natural and induced mutation through chemicals and radiation? Second part to this is, how true is the claim that gene edited products or food is as good as induced mutation?
This is a good question, which is often asked. Obviously, the whole process of radiation, or first of all, I'm assuming people are familiar with these very old and no longer used processes of chemical or radiation mutation, which were really very crude and produced actually a limited number of useful plants during the 1950s when they were employed, because they create such huge damage that the outcome was rarely of any use. Now, that obviously differs very different from gene editing induced mutations. The argument is that, well, with gene editing, it's targeted, the radiation is random. But actually, as we've seen from my presentation, there is a lot of unintended random mutation from gene editing as well as even non-random mutation from the gene editing. A lot of the unintended mutations, off-target mutations from gene editing, would be in other genes because of the way that the gene editing tool works. By recognizing a sequence of DNA, it will recognize the sequence you want, but it can recognize other similar sequences and bring about unintended damage there. So when we compare mutagenesis through radiation and chemical and gene editing, we need to consider not just quantity. Oh, you see, radiation creates greater number of unintended mutation from gene editing. It's not a quantity argument, but a quality argument as well that we need to bring into the discussion. Because when you bring this in, actually the chances of gene editing bringing about damage in another gene is greater. in another unintended gene is greater than from random mutation. And the other main difference is because of their, although it was used a long time ago, the products of mutation breeding and mutation breeding through chemical or radiation, they, of course, if we knew now about the complexities of gene structure, function, the totality of the gene network, we would question the safety of these methods as well. This is the point. But they have been there for a long time. And so even though in the European Union, for example, they say, legally say, radiation and chemical mutagenesis is a genetic modification procedure, but they do not regulate it because it has a history of safe use. And here's a major difference. These old style mutagenesis methods have had a history of safe use. But with gene editing, this is 10 years old. It has no history, let alone a history of safe use. But yet we have this huge push for deregulation. This is why it is so against scientific reasoning and scientific evidence to deregulate something that has no history of safe use, which is clearly what's happened in India and happening elsewhere in the world as well. This is a long answer, but I hope it addresses the question. There was a second question. What was it?
To that how true is the claim that genetically engineered food is as good as induced mutation?
As good as induced mutation. All I could say is that I don't think the term I'm not sure what it means by as good as. You mean in terms of bringing about the intended outcome? If that is what's being asked, yes, in theory, you could, through natural breeding, you can bring about the same outcome as gene editing can bring about something that could occur through natural breeding. That has never been proven, shown, but that is the theory, the assumption that is being proposed for deregulation. But the processes are completely and utterly different. When you do natural breeding, where you cross-pollinate two varieties of wheat or something, and you end up with new variety, that is a very different procedure than where you have this artificial laboratory procedure for genetic manipulation. As a result, the two processes bringing about a completely different, not only number, but quality of unintended DNA changes. And that is why they must be regulated differently. Obviously, natural breeding has history of safe use as well. Gene editing has no history, let alone history of safe use. Let me give you a trivial example as to how ridiculous this idea for deregulating gene editing is. Say you have electricity produced with coal-fired power station is exactly the same as electricity as you produce with solar cells. But would you regulate the two processes the same because they have the same product? Somebody with a coal-fired power station may say, I produce something that occurs naturally because there's lightning, you know, I only mimic what happens naturally. Therefore, you should deregulate me. I mean, this is, of course, ridiculous because coal-fired power stations can be highly polluting and need to be regulated to avoid this environmental pollution compared to solar cells, which produce very little pollution, and even if electricity naturally exists. So these arguments are really ridiculous. And to ignore process is being completely unscientific. The scientific process, the scientific method is the very foundation of science. So what these advocates are of ignoring process, just focus on the product, are basically they're saying, ignore the scientific method, ignore the foundation of science, just look at my end product and forget about everything else. This is how ridiculous this whole idea, this whole proposal of deregulation focused on end product and ignoring processes. And to great danger. The point is, it is highly dangerous to ignore process, because then you ignore where things can go wrong and you don't enact appropriate regulation as a result.
I will just take two questions based on this topic. Next, we'll move to Dr. Ramu. The question is, when we are talking about gene editing, we are actually talking about creating a new species. Is it true? That's one question. Second question is, could you also please explain how natural variations occur in the genes? Is it similar to the cut in the DNA with random fix, or it's a different way?
Yeah. The first one, can you just say the first one again? How
when we are talking
are we
about
creating a new species? We're not creating a new species. We're creating a new variety of a plant or animal that has a new trait. Yeah, you're bringing about a new trait, which could be resistance to a herbicide, or as I mentioned, maybe you change the composition, the oil composition, like in the soybeans, or the starch composition in the corn, the maize. So you're not bringing a new species, but a new variety, a new, which has a new trait, a new characteristic. And the second one was the about.
Could you also explain how natural variations occur in genes?
Yeah, a natural variation occurs through natural breeding, of course, because during natural mating, crossing or mating between animals or plants, pollination between plants, there is during this process of producing the sex cells, the eggs, sperm, the pollen, and the ova. These there is some what we call recombination events that brings about new variations of genes that already exist as well. But these are relatively, compared to the numbers of mutations that happen through genetic modification, they're much lower. In one very good study conducted by Chinese scientists, where they compared gene variation through, this is using rice. They looked at the number of new gene variations occurring through natural pollination of rice compared to gene editing of the rice. And the numbers of unintended or unintended mutations from the gene editing was many-fold more, much higher than in the natural pollination breeding of the rice. And but this is again not only a quantitative. I don't want us to think, oh, there's more in one version, like the gene editing, and than in natural breeding. It's not just the quantity of gene variation that's important, or gene mutation, but the quality. It turns out that naturally in the genomes of all organisms, plants, animals, humans, there are certain regions of the genome, of the DNA of the organism, that is protected from mutation. There are mechanisms in place, we don't know them all, that protect these regions from mutation. Why? Because they contain genes that are whose function is fundamental to life. And therefore, any mutation in these areas cannot be tolerated because they are incompatible with life. And therefore, there are mechanisms there to protect damage in these regions with gene. So that's why in natural breeding, the variation that occurs through natural pollination and breeding is more restricted. It could only take place in a more restricted way compared to gene editing. With gene editing, you can change any part of the genome, even the vital regions, or accidentally damage, inadvertently damage the regions that are normally protected from DNA damage. So it's the quality issue which is more important than the quantity issue here. And definitely I would say that gene editing is qualitatively far more damaging in its outcome than natural breeding.
Thank you, Dr. Michael. Thank you very much. We have nearly 10, 11 questions. Towards the end of 30 minutes, we have kept for Q&A. अभी हम next segment में जाते हैं, Indian regulation. For that, I would love to invite Dr. Raman Jayelu. Dr. Ramu is the executive director of CSA, Center for Sustainable Agriculture, and through his work, and he has helped a lot of farmers shift towards agroecological practices, thousands of farmers in India, and is working in policy level changes for the systemic food systems in India. And not taking much of your time, Dr. Ramuji. Yeah, I hand over to you. You're on mute.
Yeah. Thank you. Good evening, all. It's a wonderful presentation by Michael. And not much is left actually after an elaborate presentation has been made. But I'll take you through the regulatory framework in India and then that the changes which are being attempted to make, particularly for the genome editing part, what is happening now. So if you look at historically, in 1989, the rules are framed for regulating the genetically modified organisms. And 1998, we first discovered that illegal field trials were happening in Andhra Pradesh. 2000, illegal cultivation of BT cotton in Gujarat was found. And then 2002,
it's
BT cotton.
4:30.
I'm sorry.
In 2002, BT cotton was commercially released. So we had a history of 20 years of BT cotton in India, and which is the only commercial released crop. And 2005 is the only time when BT cotton was reviewed and officially reviewed. And then the first review resulted in ban of the 3 varieties which were released. And later on, there was no release, there is no review, even though there were several reports which claimed that BT cotton is not performing as per the claims being made when it was released. And more importantly, between 2005 and 2010, several illegal field trials from BT okra to BT maize to HT cotton were found. And 2008, first time illegal cotton cultivation, HT cotton cultivation was also found in Gujarat. And still we are finding that there is a commercial cultivation of HT cotton happening. A couple of years back, there was a study done by the Prime Minister's office, and which talks about nearly 20% of the cotton cultivation is under HT cotton. And 2010, the BT brinjal public hearing happened, and when there was an attempt to commercialize BT brinjal, moratorium was brought in at that time. And the key question which was raised during the moratorium was also about improving the regulatory system and bringing in independent lab facilities. And 2013, Government of India tried to bring in Biotechnology Regulatory Authority of India, a regulatory system, but which has not taken shape at all from then onwards. The Technical Expert Committee on GMOs appointed by Supreme Court also brought in a lot of concerns about the GMOs, the way they are regulated, and then the experiences of commercialized BT cotton as well. And 2022, now the rules are for exempting gene editing from the regulations came in. So putting all these things together, mainly we have 3 concerns. The first concern is about the validity of the claim itself. There is, even when the GM crops were introduced, the first thing we were all asking was whether it is required and whether the claim made that by the developers is right. That's nowhere they are exempted. They are not explained. And now, without looking into it, if we are even diluting the regulations, then it would be much more serious problem. Now they're talking about climate change adaptation. They're talking about several other things as part of this new technology, using the new technology. Second, as rightly Michael Antonio has mentioned, biosafety concerns still remain. How are we going to address these biosafety concerns is an important issue. Third, very important dimension of the whole problem is also about the intellectual property rights. And we have seen with the BT cotton, the kind of arm twisting the companies have done with many of the licensed companies. And then the way seed prices in India went up was very clear. And the seed companies were reorganized and then coming together, takeovers, mergers, all were happening. And now, a couple of days back, we have seen that even the 2 warring factions of the seed company associations came together. So intellectual properties rights are an important area which we also need to understand what would be its implications, and gene editing also comes under that. So when we are talking about the concerns about gene editing, we need to look from the background of the Indian experience on BT cotton, BT brinjal, and then several other genetically modified crops regulation in this country. I'm not getting into what is gene editing, which was very well explained. So the Government of India has notified new rules, issued set of guidelines, which talks about SDN 1 and SDN 2 are exempted from the regulation, under the 1989 rules. And these, they're talking about, they're exempted because there is no transgene inserted into the plant. So the only claim is that there is no transgenic. So they are not transgenics. They can be exempted. That's the only claim which is being made, and then the rules have been framed. And they're talking about CRISPR technology and other new technologies give them precision in terms of inserting the genes. So since there is no external gene inserted, and also there's a precise way it can be altered, it can be exempted. That's the claim being made. And 7 rules, from 7 to 11 of the GMO rules are exempted or not applicable for this gene editing, as new memorandum of which was released. It's about approvals and prohibitions, so production, deliberate and unintentional releases, permission and approval for certain substances which are used and produced, and permission for approval of foodstuffs. And all these 5 areas, 5 rules actually talks about controlling the biosafety of the genetically modified organisms. And now when we are taken out, all these 5 have a serious implications when they the way these would be regulating the gene edited crops. So this is what they're talking about. And the initial stages are regulated, and then with the ICA, with the RCGM approval, they can take up, and rest of the things can be done by the Institutional Biosafety Committee. Information is sufficient. It's not regulated under the GAC at all in the initial stages, and the rest of the rules apply as it is. But the issue is already, Michael Antonio has already mentioned, whether we are taking out a gene or we are making particular gene to express more or making changes at several places. There could be many things which can happen. So when we are talking about gene editing, it is not one product or one way of doing it. There could be many, many more ways of doing it. So there could be several applications of this gene editing. And that, which means it also can have different possibilities and then different implications for the biosafety. So it's not just one. So it's not about just silencing one gene or increasing the number of one particular gene. So there could be many more traits which could be altered. Even the intentional alteration could be many ways, and unintended alterations could be many more, which are not studied at all. So unless we have proper biosafety system established, it's going to be a serious issue. Similarly, though the claims are that the CRISPR-Cas and many other new technologies are very precise, unintended consequences are many, and it's already many research papers talking about those things. Already they were all presented. And more importantly, how the choices of farmers and consumers can be protected. Because once they're taken out of the GAC regulation, biosafety testing would be diluted. Also, they will not be labeled. So once the labeling is taken away, the consumer choices and the farmers' choices would be affected. So I think that's a key concern which we need to worry about. So at this point, what can be done? I think regulation should continue. We all should ask for continuing the regulation without any exemption for gene editing. and validity of the claims need to be ascertained. It cannot be left to the institutional biosafety committee alone. We have seen several examples till now. Suddenly some product comes in and then you don't know what has happened to that. Even claims are being made, no validation. I remember one GM tomato coming by from National Research Center on Plant Biotechnology, which has a long shelf life. Nobody knows where it happened, where it went, and then now even ICR won't talk about it. And last year there was a claim that non-GM herbicide tolerant rice is released, and nowhere any data is available about it in any of the reports of the ICR. So in such situation, how do we bring in institutional systems for regulating any of these claims is very, very important. Biosafety regulatory failures are seen, and we have seen even in the last 10 years, even after the moratorium has been imposed, no improvement in the regulatory systems happened in this country. So I think we need to really be worried about this one. And IPR landscape is very, very important. There would be much more implications of this because this won't even come under regulation. Earlier, if you have seen with the BT cotton or other crops, since there was this heavy regulation, the product range was also very limited. Now, if the regulation is removed, the product range will increase, which also means the complications of IPRs will increase significantly. And even if there is any exemptions, it should be specific risk. It should be very specific. So by default, regulation should not be removed. Even if they have to, if they feel, if there are any assessments need to be done, look at case-specific risk assessment, considering both the process and the product-based risks, and then look at if anything need to be analyzed, anything need to be exempted, but don't make it a blanket exemption. I think that's what we should ask for. Thank you.
Shtiyab ji.
Dr. Ramu, can you start from the first slide, please? तो डॉक्टर रामू ने पहले तो एक सरसरी निगाह से जो इतिहास है भारत में जीएमओज और उनके रेगुलेशन को लेकर के उस पर एक नज़र डाली है, और उसमें उन्होंने ये बताया कि इन तमाम चीज़ों को अनुभवों के आधार पे 3 जो मुख्य मुद्दे नज़र आते हैं। वो पहला तो है कि जो दावे किए जा रहे हैं उनकी वैलिडिटी कितनी है, वैलिड है भी कि नहीं है। ये एक बड़ी चुनौती है हमारे सामने, एक बड़ी समस्या है। दूसरी इसके बारे में भी डॉक्टर माइकल ने काफ़ी अच्छे से बताया कि जो दावे हैं प्रेसिजन के या फिर एफिकेसी के, वो दावे अभी तक सिद्ध नहीं है, क्योंकि ये एक नई टेक्नोलॉजी है। इसका अपने आप में कोई इतिहास नहीं है, 10 साल का इतिहास है। तो वैसे में इसकी सुरक्षा से जुड़े हुए दावे जो है वो कितने तार्किक और कितने विज्ञान पर आधारित हैं, इस पे सवाल है। दूसरी चीज़ है बायोसेफ्टी को लेकर के जो चिंताएं हैं वो बहुत स्वाभाविक हैं, और जो चिंता को लेकर के कि ये एक नया तरीका कहीं न पैदा हो जाए, इंटेलेक्चुअल प्रॉपर्टी राइट्स के आधार पे किसानों को उनके बीजों से बेदखल करने के लिए एक नया हथियार के तौर पे ये सामने ना आए। ये एक बड़ी चुनौती है हम सबके सामने में। वो ये भी बता रहे थे किस तरह से जो बीज को लेकर के काम करने वाले बड़े बिजनेस घराने हैं, और 2 जो सबसे बड़े थे वो अब तो साथ आ गए। तो वैसी सूरत में जो किसानों के बीज अधिकार हैं और उनके जो IPR हैं, उन पे इसका क्या असर पड़ेगा, इसको देखने की ज़रूरत है। इसके बाद जो स्लाइड है वो जीन एडिटिंग क्या है, तो उसको डॉक्टर रामू ने स्किप किया, क्योंकि इसके पहले हम लोगों ने काफ़ी सारा उसके बारे में देखा समझा था डॉक्टर माइकल से। इसके बाद जो नए रूल आए हैं 2 मार्च 2022 में, उसके बारे में उन्होंने बताया कि इसके जरिए ये जो इस नए रूल के जरिए कई तरह के यानी SDN1 और SDN2 में जो जेनेटिक एडिशन प्रोडक्ट जेनेटिकली एडिटेड प्रोडक्ट्स हैं, उनको कई तरह के रेगुलेशन से फ्री कर दिया गया है। खास करके ये कहा जाता है कि ये बायोसेफ्टी असेसमेंट के नियमों से इसको एग्जेम्प्ट कर दिया गया, यानी अब इसमें बायो सेफ्टी असेसमेंट की अब जरूरत ही नहीं। दूसरी चीज जो है कि ये दावा ये है कि इसमें कोई बाहरी ये DNA इंसर्ट नहीं किया जा रहा है, या कोई दूसरा जीन इंसर्ट नहीं किया जा रहा है। इस वजह से भी इसको ट्रांसजेनिक प्रोडक्ट नहीं कहा जा सकता है। ये जीन एडिटेड प्रोडक्ट है, ट्रांसजेनिक नहीं है। इस तरह से कई तरह के जो बहुत ज़रूरी रेगुलेशंस थे इनको लेकर के, उन रेगुलेशंस को कमज़ोर और हटाया गया है। नेक्स्ट या सो इसमें जो बता रहे हैं कि जो रूल है 7 से 11 तक 1989 का जो रूल है, उसके अंदर जो सब रूल हैं 7 से 11 तक, उनको हटाया गया है। उनको कम से कम इस मामले में SDN1 और SDN2 के मामले में उनको ड्रॉप कर दिया गया है, हटा दिया गया है। तो अब आपको अप्रूवल और प्रोहिबिशन आदि से जुड़ी हुई चीज़ें, या उनके उत्पादन से जुड़ी हुई चीज़ें, या उनके यानी इरादतन या गैर इरादतन रिलीज से जुड़ी हुई जो मामले हैं, इन तमाम चीज़ों को अब रेगुलेशन के दायरे से बाहर कर दिया गया है। यानी उसको कम से कम इन दोनों ट्रेट वाले जो जीन एडिटेड प्रोडक्ट्स हैं उनके लिए। नेक्स्ट स्लाइड। हाँ, तो पहले जिस तरह से प्रक्रिया को लेकर के रेगुलेशन था, वो अब नहीं किया जाएगा, है ना, नए रूल के आने के बाद से। नेक्स्ट। हाँ, तो डॉक्टर रामू का बहुत साफ तौर पे ये कहना है कि जो इसमें कुछ चीजें बहुत ही टेक्निकल है, वो मेरी समझ के भी बाहर है।
नहीं, मैं ये बता रहा हूँ कि अगर पूरा जिम्मेदारी अगर इंस्टीट्यूट बायोसेफ्टी कमेटी को दिया गया, तो अभी तक जो GE से रेगुलेट कर रही है वो भी ठीक से नहीं कर रही है, और वो निकाल के सिर्फ IBSE को दिया तो ये बहुत मुश्किल हो जाएगा। दैट्स द ओनली पॉइंट। You cannot leave entire thing to institutional biosafety committee alone as per the new rules.
तो नए जो नियम है उसमें पूरा का पूरा पूरी जिम्मेदारी जो है GEC को चूंकि इससे अलग कर दिया गया है, तो रेगुलेशन पे और मॉनिटरिंग पे उसका बहुत गलत असर पड़ने का खतरा है। तो इसमें जो चुनौतियां हैं वो यही है कि हाँ, यानी जो
इसका इम्प्लीकेशंस मल्टीपल है। ऐसा नहीं है कि जैसा क्लेम कर रही है कि सिर्फ एक ही जीन को हम मॉडिफाई करेंगे, वो नेचुरल की ऑलमोस्ट इट्स लाइक नेचुरल, ऐसा नहीं है। बहुत सारे चेंजेस हो सकता है जो जीन एडिटिंग की वजह से। इंटेंडेड चेंजेस भी बहुत सारे हो सकते हैं, अनइंटेंडेड चेंजेस और बहुत सारे भी हो सकते हैं। तो इसलिए सेफ्टी, बायोसेफ्टी टेस्टिंग के बाहर ये निकलना हमको सहमत नहीं है। दूसरा जो प्रिसिशन क्लेम है, the two claims, एक है there is no much changes done since there is no gene inserted, it's only inside the plant we are changing। तो हम पौधे के अंदर ही चेंज कर रहे हैं, बाहर से कुछ नहीं ला रही है, बट व्हाट इज हैपनिंग इनसाइड आल्सो कुड बी very severe। Second is the precision claim is also wrong. There are a number of studies which show that it is not so precise as claimed। And third point I was saying is कंज्यूमर का, किसानों का चॉइसेस क्या होगा। जब आप रेगुलेशन से बाहर निकलेंगे, उसके ऊपर लेबल भी नहीं होगा, फार्मर का चॉइस इफेक्ट होगा, कंज्यूमर का चॉइस भी इफेक्ट होगा।
Thank you, Dr.
Yeah.
Okay. I think there's one thing. Yeah, please.
So the last.
हाँ, इसमें इनका कहना है कि जो रेगुलेशन अभी तक रहे हैं उनको बरकरार रखना चाहिए, और ये जो भी दावे किए जा रहे हैं उनको केस बाय केस उनकी वैलिडिटी या उनकी जो प्रामाणिकता है उसकी जांच कराने की ज़रूरत है। और बायोसेफ्टी रेगुलेटरी फेलियर्स और जो चिंताएं हैं उनको यानी प्लग इन करने की ज़रूरत है, उनको दुरुस्त करने की ज़रूरत है। और इंटेलेक्चुअल प्रॉपर्टी राइट्स के जो मामले हैं उनको बहुत ध्यान से देखने की ज़रूरत है और उनको समझने की भी ज़रूरत है, और उन पे ख़ास तौर पे फोकस करने की ज़रूरत है, क्योंकि अगर उसको छोड़ा जाएगा तो किसानों के लिए काफ़ी वो प्रॉब्लमेटिक एक बात सामने आ सकती है।
Thank you, Dr. Ramu, for giving us such a brief history and the regulations and what can we do in such a liquid manner. Hello, can you hear me?
Yeah.
Yes.
Yeah. So I will now open up the floor for Q&A. I'm really sorry that we kept unmuted for all of you. So one by one, you can raise hands and you can take up the questions. Yeah, you will be able to unmute. So I will just take names in the order. So Sri Harsha, then Selvam, then Aruna. If you, Sri Harsha. This floor is open now. If Sri Harsha is not available, Selvam.
Hello.
Sri Harsha, please go ahead with your question.
Yeah. So I was in one of this conservation science meeting, I think so 3-4 years back, and one of these scientists were kind of sharing about CRISPR and how it can actually be used for conservation, and so I actually did raise this question. I think even Jairam Ramesh was in the crowd, and I did raise this question, but I got a very I got a similar answer that it is precise, and it is not like the GM kind of a thing in itself. I just wanted to understand. So this is a moral dilemma also, right? When they can bring it in the guise of saying, because a species is getting extinct, we will, use this as a front, and this is an opportunity. So in the wildlife conservation space, so I was just thinking of how is it right in that space, and are they using that as a front, to push this through also, because a lot of crops would also be extinct otherwise, also a lot of species. So that was the context I wanted to raise this issue.
Michael, you want to answer this?
I'm not sure how gene editing can be used to prevent extinction. since the problem is not a genetic one at all, but one of environmental conservation. What this kind of mindset illustrates generally how genetics is being used, environmental problems are being used as an excuse to push what they would call a genetic solution. But actually, but since the problem is not one of genetics, I fail to see how a genetic manipulation of 1 or 2 genes is going to help at all. So I find this again very disingenuous, to be honest, and I see no use. I mean, to be honest, just to add to this briefly now, the problems we face in food production, if we go back to that, and environment, are system problems. Yes, they're not. There's nothing wrong with the crops and animals we have now. There's nothing, and when it comes to environment, it's human degradation of the environment that is the problem. So these system problems don't have a genetic basis. So offering a kind of genetic solution by, say, tweaking a gene or 2 in a plant or an animal, or in the environmental context, I'm even more at a loss to see how it can be of any advantage. I find it being completely detracting from the real causes, detracting attention from real causes of the problem, which are system problems, and introducing these things are there in a way to maintain an intense chemical-based, unsustainable, failing agricultural practice. This is what it's all about. In England, many years ago, the term sustainable intensification was coined by the British government agencies. What can we do to maintain a system of intense agriculture that is damaging anyway? How can we, which they recognize as unsustainable, but how can we sustain intensification, which by definition is unsustainable? For me, this is a contradiction in terms. So we need to put in place appropriate conservation for the environment to prevent degradation. And in an agricultural context, we need to put in place sustainable agroecological systems. Yeah, the problem is a system one. The claims that gene editing will produce crops that are drought tolerant, stress tolerant, pathogen tolerant, disease resistant, and so on and so forth. These for me are fantasy, because these traits are not on the basis of one gene. They're on the basis of the holistic functioning of the totality of the genes of the organism. Yeah, genes are like an ecosystem, an interconnected ecosystem, just like the environment. We can draw parallels, and changing one thing in the ecosystem can have major repercussions because of the interconnectedness of the system. So this is it really. You will fail to produce robust, drought-tolerant, disease-resistant, pathogen-resistant, etc. plants or animals. You will fail because these are what we call complex traits. This addresses one of the questions I saw in the chat. They are complex traits. In humans, a complex trait would be our height. Yeah, I'm relatively short. Maybe some of you are no doubt much taller than me. The reason why is because it's many gene families and the whole genome functioning together as a whole that determines a complex genetic trait such as height. And it's the same in plants. We have complex traits that are there because of totality of the functioning of the organism. And changing one gene or another in a plant or an animal is not going to solve a system problem. A complex trait will only be introduced if you bring together all of the genes all the gene families that are needed for the complex trait, such as drought tolerance or heat stress resistance. Only the totality of genes working together as an integrated family network will work. Tweaking one or two genes here and there, doomed to failure. So I warn you, don't get taken in by all these claims that we need gene editing to modify our plants to meet the challenges of climate change. No, this is not going to work. It'll be a total waste of time and money. Or change the system. We need climate-ready systems, climate-ready agroecological systems to move forward to meeting our food needs now and in the future. Not gene-edited plants grown in an intense chemical-based, unsustainable practice. This is, a waste
Just
of
to
time.
add on two more points. The whole idea of conservation is about maintaining the existing thing. So gene editing actually intends to change the existing one itself. So it's a contradiction by itself that it will aid in conservation, because you are trying to modify and then it won't result in any conservation. Second important thing, the moment any trait has to express, it depends on the environment as well. So just by making genetic change, it won't it won't show the result. It will be based on the environment. And the whole worry is that environment is degrading. So if you are not making any change in the environment, any change in genetics will not help. And if you can make the changes in the environment, the existing variants in the environment, the genetic pool is sufficient to meet the needs. Many of the crops, many of the varieties are failing because they are unable to express themselves with the changing climate. So I think that's these contradictions in the claims we need to precisely understand and then present it when such. See, in India, what we are seeing is they simply use jargons and then get away. The claims are made. Claims, they just make the claims and then get away. I think this is very important to have such clarity, which we need to talk about all this. Yeah.
Yeah, very good. Yes, very, very good.
Selvam?
Yes, there are a few questions. So one is, usually, can you hear me?
Yes.
Yes.
Hello? Okay. Usually when high-risk technology or high-risk product is introduced, regulation is coming in, and immediately the lobby for the product or the technology works, the system, government system, how the regulations should be, what are the protocols, test protocols should be like that. Here in GMO, what are the tests really to be done, and what the lobby is working has made in the protocol systems, what kind of a test they actually avoid to get through the permission. This is question number one. Then question number two is, Antonio Michael has said gene is working as a network, or gene is a network of genome is a network, and every gene is connected with that. Gene's expression or gene functioning is also part of the network. Is there any easy example to say to a common man how this network gene is working along with other genes as a network part of it? This for to make understand the people easy, common people easy. Then can we say genome when naturally, normally the people say that one gene is responsible for one product, one protein or things. But can we say that a gene has hidden responses that we don't know? Or what we know is a gene is responsible for a particular protein. And what we don't know is a gene is unknown for unknown when this comes an agent. Is there such a kind of a thing is there? Thank you.
I have sent the question again in the chat box.
Yeah. So in terms of regulation, like I mentioned earlier, the institutional biosafety committee is empowered to take the regulatory system. So they need not go to the GEAC. That's what the new law says. But the key issue, while such tests are prescribed or tests are done or the decision is taken, these are all done by the Department of Biotechnology, which is a promoter of such technology itself. I think that's a contradiction by itself. That was the ask we were making for a long time. How can a promoter be a regulator? How can promoter be a regulator? And they are the opinion makers as well. So I think these contradictions need to be addressed, and that's what Jairam Ramesh in his moratorium clearly mentioned. So we should have an independent system, and then it should be, even testing should be independent. So that is never addressed in this country. So the concerns raised 10 years back, and in the last 10 years, whatever the problems we have seen, they were never addressed. And then now we are saying even the weakest system which is there is not required. I think that's the problem. And the rest of the transgenic technologies and all, are all already outdated. Companies are already moving out of it. So there is no point in, I don't think any new technology, new product will come in from the transgenics for any approval also. So now most of it which is in the pipeline are all about gene editing. And then now if you are removing from regulation, it's going to be a serious problem. So this is something which we need to be worried about. The second question probably Michael can answer.
Yes, thank you. Just to add, the danger of you ask Selvam about what testing protocols will be required, but the point about exemption and deregulation is that no testing will be required for safety evaluation. Like what's proposed here in the UK is it would simply be a reporting system. Anybody can produce a gene edited product, which if it feels meets the criteria of the government, all it has to do is inform. It's simply a registration informing system. There'll be no need for any risk assessment to health and the environment, and there will be no requirement for labeling and monitoring after launching on the market. And this is the danger we face, is a total lack of safety testing and monitoring. And in terms of the concept of genes working in networks, it's a generally accepted concept. I tried to illustrate that by using the whole idea of complex traits and in humans something like height and other complex characteristics, which could be the general health status or disease status. We also have complex disease states, which could be cardiovascular, they could be psychiatric problems. We now know these are complex traits which have at their basis a dysfunctioning of the entire DNA, the entire genes of the individual. And so complex traits in plants will include things like, and height, and normal things, I'll mention height. Now in plants, complex traits are like yield, responses to environmental stresses, resistance to pathogen, infection by fungi, blight, or even resistance to attack by pests. These are complex traits again, which will have, so when a plant gets infected, or the yield of the plant is determined by the functioning of the totality of the whole plant and how it interacts with its environment. It's not down to one or two genes. So all these claims, oh, we changed this gene and you see, wow, amazing, we get this, we will get massive more yield. No, that's just not going to, it won't happen and it won't be robust. For example, in England, one of the things that's being on development here in the UK is gene-edited pigs that will be resistant to infection by a certain respiratory virus, which is common in pigs because why? Because they're kept inhumanely so packed together that the virus just, the pigs are unhealthy and the virus spreads easily. So they want to gene edit a pig so that the virus cannot infect the pig anymore. How long will it take for the virus to adapt to overcome a one gene block? How long? I would say it will begin immediately because microorganisms change their genetic characteristics incredibly rapidly, especially viruses. We've seen that through the through COVID. How long did it take for new variants of SARS-CoV-2 to appear? Straight away. And therefore, any efficacy that may be there from vaccines was very, very quickly lost. So this is how conceptually flawed. It's very conceptually flawed. This whole game of GM and gene editing, it's totally conceptually flawed. It goes completely against contemporary understanding of molecular genetics, how genes are organized and how they function in an interconnected network. And then the products as well. It's not just at the level of the genes, but at the level of their products as well, the protein products, and their biochemistry and so on and so forth. No entity works in isolation. This is the fun thing. And the whole is greater than the sum of the parts. You change one part and you can have major ramifications on the whole in an undesirable way.
Aruna, you're muted. Please unmute.
Michael, I would just like to remind you that there are 3 or 4 questions that are pending for you to talk about already, which I sent by email.
Yeah, if you can remind me, I will deal with it.
I will remind you later. In this conversation, which you have just presented, I have 3 questions which are puzzling me. But before I come to that, I just want to remind everybody that Ramu went through some of the history of GMOs in India. I just want to say, as far as the Supreme Court is concerned, we are at the stage where we are talking about a moratorium. We've been through the technical expert committee report where the first question, do we need the GMO, has been struck down conclusively. We don't need GMOs. We are asking for a moratorium. On the gene edit products, we are saying they are within the 1989 rules. The government is deliberately lying. That's going to be the process in the court. And we need to stick to our point of view, moratorium, including on genetic edited products. We don't go into regulation. It doesn't work for many, many reasons, as well as because regulation is not amenable to translating what unintended effects there might be. So we are asking for a moratorium. Now, the 3 questions I have, which are puzzling me. Oh yes, there is one more point. The central plaque of the moratorium is contamination, because it will change our agriculture, our food, and our feed. I think the same thing applies to gene-edited products. Am I right, Michael? Contamination applies to, it's exactly the same as GMO.
Exactly the same, because it is possible if with 2 things, with law obliging the gene editing product developer to disclose, an obligation to disclose the gene edited change and provide reference material, it is very straightforward to devise a test to detect a given gene editing event. And therefore, it's possible to look for any contamination of that product. Therefore, the same rules should apply. I know this is a good point, Aruna, because one of the arguments that's been made is, well, you wouldn't be able to tell the difference if the gene editing event happened naturally, and that's why it's in your mixture of wheat, rice, beans, whatever, or whether it's actually the gene editing event that's been come from the laboratory. But actually, it is possible. It is very straightforward to develop unequivocal tests, because the gene editing event will be in a given genomic context that will be unique to that gene edited variety, that will be very different to any plant that may happen to have the same gene editing, the same genetic alteration that happened naturally, which is probably a near impossibility anyway.
Okay. The three questions that I have, Michael or Ramu. One is, you made a reference to the tool being actually inserted into the plant. I don't understand this. Are you saying that the insertion of CRISPR-Cas is therefore an internally, it is internally, it functions as an internal process and the engineer has nothing to do with it?
Yeah, okay.
That's my first question, yeah.
Yeah, let's deal with this. Again, good question. There are two ways in which the gene editing tool, the CRISPR-Cas tool, is introduced into the plant. There are two ways in which it can be introduced. One is as a pre-assembled complex of protein and guide RNA, which can be introduced, and then it does its edit. And because it's just protein and RNA, it quickly breaks down and will be, no genetic material will, or protein, nothing will remain behind. Now, this is actually very, very difficult to do in plants, extremely difficult, which is why, to this day, virtually all of the gene editing of plants is done by introducing not the pre-assembled CRISPR-Cas guide RNA, but genes, plasmids, which have the genes that code for the CRISPR-Cas and the guide RNA. Okay, so these are plasmids, circular DNA molecules. I illustrated that. I tried to illustrate that in one of my diagrams. Now, again, after you've introduced the plasmids encoding the gene editing tool, the CRISPR-Cas on this occasion, into the plant cells on the dish in the laboratory, there are two possible outcomes that you can engineer. One is that the plasmids can insert into the DNA of the plant cells and express from there the gene editing tool to bring about the gene editing event. The other is where you engineer it so that the plasmid does not splice into the DNA of the plants. It remains separate, but still expresses the genes for the CRISPR-Cas gene editing tool to bring about the event. Now, in the past, it was gene editors used to integrate the gene editing tool and then bred it out. After they got the gene editing event, they would then cross-pollinate, back-cross to the parent plant and breed it out. But that is not done so much now. What's done now is the plasmid encoding the gene editing tool is introduced in such a way that it does not have such a great propensity to splice into the DNA of the plant cells, but remains separate, but nevertheless expresses the tool and brings about the gene editing event. And you would think, well, that's all right, isn't it? Because the gene editing tool, the plasmid, the DNA of the plasmid will eventually be lost after you grow up the cells to a whole plant. But that, as I mentioned in one of my slides, that is not always the case. In fact, almost invariably what happens is, and this can happen with either of these two plasmid-mediated transformations, is that the plasmids can fragment, and fragments of this can splice into the DNA at random around the DNA of the plant. You can have these fragments of the plasmids splicing in to the genome. So actually you can end up with a gene-edited plant following an SDN1 or SDN2 procedure that does contain foreign DNA, which would be missed unless you oblige the developer to conduct a thorough, in-depth whole genome sequencing analysis, which at the moment, there is no obligation to do so at all. And this has been illustrated in plants and also illustrated in animals. I illustrated to you that there's been more than one example in animals where the part of the gene editing DNA that was introduced into the plant, into the animal cells, the whole plasmids got spliced into the DNA of the, in this case, the cow, the calf, the cow DNA from which the cars were produced. So it's a very well documented outcome. And so, and we have to remember the law, not just in India that is proposed, but elsewhere, is that the final gene edited product must not contain foreign DNA. But I can almost guarantee you that if you did a proper in-depth genetic analysis, you will find foreign DNA there, even though it was not intended to be there. And so this is part of the, you know, there has to be an obligation to do the right analysis, which at the moment there is no obligation to do so.
Thank you, Michael. That was exhilarating. I didn't quite understand all that. I'm a little clearer now. My second question is with regard to RNA.
Yes.
Another very blurry issue as far as I'm concerned, because I know that RNA from eating plants can interact with our DNA.
Yes.
So now what happens with genetically edited products? The amount of RNA that they can produce is something that is inherently dangerous. Am I right?
Well, I think here you're referring to the guide RNA of the CRISPR-Cas gene editing tool.
Yes, yes.
Which is crucial. You know, it's an essential element to target the CRISPR-Cas complex to the region of the organism's DNA to bring about the edit that you are seeking. But you know, the crucial thing here is that guide RNA may not just recognize the region you're trying to target, but may also recognize similar sequences elsewhere in the DNA of the organism, whether it's plant or animal, and bring about an unintended DNA change or mutation, in addition to any intended mutation that you are trying to introduce. So it's very different. The kinds of RNA in foods that you're referring to that could affect our gene function and therefore our biochemistry and health status, or what are known as microRNAs, they have a very different function and mechanism of action. And so therefore, we cannot say that the guide RNA will have something akin to what this microRNA mediated gene regulation may take place through us ingesting microRNAs that are present in our foodstuffs. You're right, these microRNAs are present in foodstuffs, and when we ingest them, they do enter our bodies. And in principle, they could pick up similar gene sequences and regulate their function. But this is very different to the RNA element that's part of the CRISPR-Cas complex. I do not see it that has its own complications, but they're different to the complications that may arise from especially GM plants that have been engineered to produce a microRNA as an insecticide, for example, or plants that have been sprayed with a microRNA insecticidal spray.
So, Michael, are we saying that the RNA generated for gene-edited products is not you said it could be dangerous. It can't interact with our DNA? Are you saying it
only? Yeah. It's not the way it interacts. As I say, it acts as a guide. The sequence of the guide RNA of the CRISPR-Cas recognizes and binds to DNA sequences of the sequences of DNA that are similar, and it's designed to go to one area. But of course, it can recognize similar sequences other than the intended site and bring about an unintended gene edit or off-target. This is how off-target edits take place.
So we have a problem. We have a problem.
There's a problem. There is a problem there of off-target DNA damage.
Right. Okay.
But not of the kind that arises, say, from the naturally existing microRNAs or engineered microRNAs that may be in GM foods or in insecticidal sprays.
Okay, I'll pursue that separately as far as the Supreme Court is concerned. because I still am rather blurry about this. My last question is with regard to plant tissue culture.
Yes.
This is not necessarily to do with gene-edited products because they are common to GMOs. I didn't realize that there were unintended mutations in the very process of plant tissue culture.
Correct.
This is new to me. I didn't know this.
Yeah. No, this has been known. You're right. This has been known for decades now. And the numbers of just growing the plant cells in tissue culture can bring about hundreds or thousands of different types of mutations, small and large, in the cell's DNA. And tissue culture at the moment is an obligatory part of not just old style GM, but new style gene editing. And so these mutations from just the tissue culture will be carried through to the final product. Some could be bred out by crossing your gene editing event back to the parent, but not all of them. The point is that many unintended, just tissue culture induced mutations will be carried through to the final marketed product that could affect the function of multiple genes with unknown, unpredictable compositional and health consequences for the consumer. So that, as I say when I'm in my talk, at all of the steps of the GM or gene editing process, there's an accumulation of unintended DNA damage, which at the end can be hundreds or thousands of sites of unintended damage that can range from just small DNA-based unit changes, few bases, to large rearrangements.
Now, to the 4 questions that I sent you by email, I don't want to take up any more time because there are people here who want to ask questions. Will you deal with that in an email to me, Michael? They deal with regulation. They deal with the obfuscating quality of CRISPR in regulation, that we can't really tell what's going on. And which brings me back to the point that we don't want, we've gone beyond that point. We don't want regulation, especially the way it is carried out in all countries. We want a moratorium and we stick to that.
Yeah, I would agree. If you can get a moratorium, that would be ideal. Because
we have to push for it on GMO, and therefore it automatically must happen for gene-edited products.
Yeah.
That is what's going on.
You don't have to push them.
Yes. Yeah.
Don't let them get away with ignoring process. The way they get away with things is by ignoring process.
Yes, yes.
You nail them, back them into a corner to take into account process. They cannot deny that gene editing is a GM process, and therefore the products must remain under current rules, which if it's moratorium, there must be moratorium or whatever. This is how they're trying to get away. You know, in all of my talks with if there's advocates in audiences, never has it been denied, even by the advocates of gene editing, that gene editing is a GM process. They cannot deny that. But so how do they do that? They try to ignore it. They try to simply focus you on the end product and ignore the process, which, as I said, is completely unscientific. And it's actually, it's dishonest, completely dishonest. And it's shameful that my fellow scientists would ever take up such a position.
Thank you. Yeah.
Next, the sequence of the next questions are from Ray, Nagarjan, and then Rosamma. And before that, I have a small request. We will extend this for 5-10 minutes more because after the Q&A, I would love this group to focus on the next steps. So please don't leave. Please stay for 10 more minutes. Ray, please go ahead.
Good evening. Thank you very much for this session. My question is related to diet and nutrition. I have mistakenly understood that genetically engineered rice, for example, rice would be something once we consume is going to break down, like carbs are going to break down to glucose, proteins will break down to amino acids. Every aspect of the food is broken down and digested or excreted. So how is genetic engineering going to have a direct impact on the human body if the human body is designed to break down these compounds and digest or excrete them. Are you saying that there's a possibility that the microRNA could, bind itself to the body and probably trigger mutations and tumors? Is that what you're saying? Thank you.
Thank you, Ray. Because of the health consequences from GM, old style transgenic GM, and new style gene edited products can arise at two levels. One is even from the intended change. If you change a protein structure that's in the food, it can now suddenly take on, for example, allergic properties. Or if it's an enzyme, it can begin to carry out biochemical reactions that it never did before and risks producing a new toxin. But over and above that is the general alteration. It's inevitable. It's not as if it's not that you may get changes in unintended patterns, changes in gene patterns in plants. They will happen. It's inevitable. It's a question of what they are and what the consequences are of the alterations in the gene functions and what effect that will have on the biochemistry of the plant, and whether that altered biochemistry includes production of a novel toxin or a novel or a new allergen that previously was not present, or increased levels of toxins that were there but maybe very, very low, but now the known toxins and allergens may suddenly increase in their amount. So this is where the sources of concern are. So yes, you may digest the rice and so on, but there may be a toxin there, or one of the proteins, the new proteins there, altered protein, may now be an allergen. And so these are issues that are largely will be ignored. If things become deregulated, there will be no risk assessment of these possibilities. And so a product can be launched and people may start suffering before down the line, and without labeling it could be difficult to then trace the source. So this is where the sources of concern are, Ray. I hope that answers your question. And there are, and I should add, there are animal feeding studies. We don't have any animal feeding studies that have assessed risk for gene-edited plants, but with first-generation transgenic plants, whether in maize, soy, especially, animal feeding studies have shown toxic effects, especially affecting liver toxicity, liver and kidney function, and digestive system and immune system problems, including allergic type reactions from the consumption of these first-generation transgenic GM foods. So there is some precedent there to give us cause for concern.
So it would make sense to have clinical trials with such foods, the way we do with vaccines and drugs.
Ideally, yes, you need to have preclinical testing in animals. And then ideally, you then need to move forward. Oh, this is what we were advocating in the 90s, actually. Not just animal studies initially, but also move forward to some feeding studies in human subjects.
Thank you.
Thank you. Nagarajan. Sorry, Nagarajan.
Yeah, thank you for this session. I'm Nagarajan. I think my question, some of it has come down to, in the explanation, I've understood that now in the final stages, moratorium is the end. But I would like to ask Dr. Michael that why can't the other group of scientists provide a lot of evidence and see, these are all assumptions. What he was explaining now also, there is chances of gene edited product can cause certain things. So this accumulation of data can be another way of lobbying or advocacy, and also this can be as a proof for many other things. Why the group of scientists or the European Union for Social and Responsibility, is there a work, legitimate and scientific work, and the data is being provided to the legalities where this can be another parallel way of suggesting this is what is happening? Enough evidences and proofs are being created from the other scientific community? That's my question.
if I understand correctly, yeah, I think I understand your point correctly, and it's well taken. First, I mean, I find the developers of these products, for me, they are very arrogantly, I would say, they think they are so in control. They're so predict all that they can do that they don't feel any need to do any in-depth analysis, including toxicity tests, to prove the safety of their product. And since the law, as things are going, will not oblige them to do that, they're not going to bother. This work can be done. It may cost quite a bit of money and take quite a lot of time, which is another reason why they will not undertake this kind of work. That doesn't mean, but of course it's needed. But somehow the developers have managed to persuade the regulators that this kind of safety evaluation and testing is not needed. And because they can just predict, they know what they're doing and they can predict all the outcomes, whereas the science tells us a completely different story. So I don't know, the solution has to come back to a change in mindset of the regulators and governments. And for me, unfortunately, the problem that we're faced with, because I find no scientific reason for not doing the safety testing, because because you need to be true to the science, and the science tells you need to do all these analyses and so on and so forth to prove something worthwhile and safe. But they're ignoring the science. And so you have to find a non-scientific reason for why they're not doing it. And for me, it's a question of, as was alluded to by Ramanjana, which has to do with commercial reasons. These products are patentable and therefore they're potentially very lucrative if they're once marketed. And because they're patentable, they give total control of the developer to how these seeds are grown and marketed and so forth. And it will make rich financial rewards for developers and for government. And I'm afraid that this is really, for me, what's driving this whole development. Because in look around, it's unnecessary. But because the patenting, following the model of the first generation GM crops, the transgenic GM crops, these are patented products and made large sums of money for companies like Monsanto and Syngenta and Corteva. And it's the same with the gene editing. The same mindset is behind gene editing. More than that, patenting the product and looking for massive financial gain. So I don't have an easy answer to your question, because the mindset of the people in power is not as it should be. It's not true to the science base. And until that changes, then we're faced with this issue that what you ask for is just not going to happen.
Okay, thank you, Dr.
Rosamma?
I was just, quite chilled by, the last answer, because so much of it is what we hear about the vaccine as well. And then now we're seeing this concerted push for changing GM regulations across the world. And we've seen similar push for vaccination. So could these all be connected?
Possibly. It's certainly, as I say, there are many that profited from the launch of the COVID vaccine-like agents. And for me, it's the same kind of industrial mindset is at the basis here as well. I don't know, that's as much as really I can say. Anybody else want to comment?
So if there's nothing much, so should we?
Just one comment, if I may. It's the same lack of science in both. We have a complete breakdown in science of the COVID policies and the vaccines. And we've got a breakdown of science in GMO and genetic edited products.
Yeah, that would appear to be the case.
Okay. So since the time is less, so since the group is diverse, so I would love to give 10 seconds each to each of you to just share what about the ideas, what can be done next. What should be focusing as a group to next? Any 2-3 ideas, one idea. Each person can take 10 seconds and just share quickly. Anyone would love to volunteer to go first?
Hi, can I go since I
Yes.
I have to shut the computer and
Yes.
leave? I think it's considering the political and economic structure of how India is going right now. It is very difficult for us to fight it, and I can say that activists are within themselves very divided when it comes to these issues. Always divided on the terms of ideology and whatnot. I think the GE is going to go ahead. What we will have to do is find solutions how to manage it. So if clinical trials is one way, one possible solution, then we should push for this rather than saying no, because I think the world looks at us and thinks that we're backward and we're very stupid to resist the future of science when we say that we don't want GE. So let's say that okay, fine. If you want to go ahead with GE, fine, but let's enforce more of the safety and safety systems. I think a lot of good information was shared today, and that could be used as part of it. Thank you.
Thank you. Next. Bunny Chakraborty. Yeah. You need to unmute. Mr. Bunny.
Yeah. Not Mr.
Sorry, Miss Bunny.
No, it's okay. It's okay. Actually, a lot of things are already told. One of the question has come to my mind, that is, we are apprehending there will be some ill effects on the health, on human, animals, and the ecological, total environmental system rather from this genetically modified crops. I want to know, it may be Michael or Amanajulu, whoever may answer it, that there are a lot of GMO crops now, sunflower, soya, and mushrooms and many other crops are already being produced by the different companies. We are selling the seeds, and the production is going on, and the crops are being exported from US and some other countries. So who are the consumers of these products or the crops, and is there no morbidity pattern changes among the consumers who are consuming these crops, whether the consumers directly the human being or the animals or any bioengineering industries? I exactly don't know. I tried to search it out, whatever the morbidity pattern changes among the consumers. Who are the consumers, major consumers of these crops? And if the not human being, if the animals, what are the morbidity changes have taken place? And as a chain effect of that, what has happened on the environmental surroundings? This is a basic question to me. So can anyone answer to this? Or is there any possibility to do that morbidity studies by anyone, any researcher, any scientist? And if so, then why it's not being done till now? When there is a lot of concerns raised in different literatures and by the scientists on different fronts, why not these studies are taken? This is my basic question to know.
So quickly to say, these are not apprehensions. Basically, the concerns are based on evidences.
Right. Exactly.
So there's a lot of data which is available, and that's one of the reasons we are asking for a regulation. Don't exempt. continue the regulation. Whatever is the regulation is there, don't exempt, but continue with the regulation, establish the biosafety is the ask we are making. I think that's a key thing. Second important thing is, in fact, the assumptions are made by the scientists who are pushing for genetic editing, basically saying it is almost equal. So assumptions are not made by people who are having concerns, but the people who are actually developing it. The assumptions is that it is almost equal. It's an assumption. Yeah. They are not established. They are not established. So they need to establish that part.
Yeah.
May I just get in once? My name is Kavita and Ms. Chakraborty. The issue is that in good science and in good scientific regulation, you're supposed to pick up early warnings. You don't wait for morbidity studies.
No, I understand.
After something is released.
Definitely so.
So in terms of evidence that exists out there, from experimental studies or from simple things, even at a molecular level, even without feeding studies and so on, of what genome editing is causing in terms of large-scale unintended mutations and so on. There are there's an entire body of evidence and very fast emerging evidence. I mean, just in the last 2 years or so, there's so many published papers that show that, this is not the precise technology that it is being projected as. Therefore, to, rely on morbidity studies to prove our point would be somewhat unwise. You begin by picking up early warnings, and they exist. And these are published scientific papers. If you're interested, can you just leave your email address? And, Ajay Tikala and some others will share such material.
Yes.
Yes.
There are sites which
I'm interested. Definitely, I'm interested to.
So we will send those. And we've been engaging with our regulator with all this material, but they still go ahead with what they want to, because the ones.
No, I'm not. I don't have any objections in this respect. I'm too concerned about this. My question is, as the products are already in the market, there's a market share has already high. That is
but you realize how complicated and next to impossible it will be to prove cause and effect in real life situation, when out there's so many factors that are affecting the safety of our food.
Of course.
So that is the reason why you have to rely on experimental studies, which are part of regulation. Regulation has to prescribe comprehensive long-term studies, as well as be influenced by evidence that is emerging out there. Whether the regulatory regime has asked the crop developer to do some tests or not. Thanks, Ajay, for giving me the chance.
Okay, thank you so much.
So I think the time is already up. If some comments are remaining, I'll just let 2 or 3 people to just give quick opinions or what should be done and what are they doing, and take 1-2 minutes more. And I will collate all this and send it on email. Selvam, yeah. Selvam, please go ahead.
The thing is, we have, the thing is, making up very much awareness, giving very much awareness to many people is one of the important things that we do keep on doing it, but there is not sufficient enough. That's the other fact. The recent political trend is coalitions. In the elections, elections will be fought with coalitions. Coalitions means there will be more minor parties in the coalition. We have to work with the minor party leaders or the policymakers or the managers of the minor parties to make awareness, educate the issue. When just more, we work in Tamil Nadu also. So it is very much important. We always keep on working with the major political party or the ruling party, are supposed to be the next ruling party. We leave out the minor parties. We have to work with the minor parties. Third thing is most of our people, aware people, simply post in WhatsApp or Facebook like that. They should write. We should make them to write to the concerned minister or the concerned responsible person in the government to know that what the public mood is. It's also very much important. Thank you. Over.
Anyone else? Or okay. I would like to thank Dr. Ramu Garu, Dr. Michael Antonio for taking so much time out in such a short notice. I just wrote them 1 week back, and thank you very much, and explaining such a difficult and complex topic to all of us in such a liquid manner. And Dr. and Ishtiaq Ahmed for explaining it in Hindi, and it was really difficult, complex job, but he did it very smoothly and we could understand it. And all of you for sitting 2-3 hours of this, and thank you very much. And Rohit, Nachiket, Kavita Garu, Kapil, and all of them who have helped me in background, and everyone who sent messages and everything. Thank you very much. Thank you.
Thank you.
Thank you.
Thank you. It was a pleasure
Thank
to
you.
be part of this. Bye. Do get in. If you need my help any further, I'm here to help.
Sure. Thank you.
Bye.
Thank you all. Thank you all.
Thank you.
I think you have a copy of the presentation, I assume.
Dr.
Some
Michael.
people were asking for it. Both
Yes,
the
I will.
presentations. Yeah.
Thank you, Dr. Michael. You simplified a very complicated subject. Thank you.
Thank you, Ray. Yes, it was a great pleasure.
Thank you, Dr. Antonio.
Thank you, Kavita.
Ajay मिल गए? Ajay, did you find the option?
No, it's not coming. Save in the chat.
On bottom right, there is usually something.
Yeah, right. Okay, I'm just copying it manually.
Fine. Okay, you stop the recording. Recording is still going on. The live streaming. Stop it before you end the meeting.
Okay.
It's still going on, huh? Ajay? Live streaming is still going on, huh?
What?
Live streaming अभी भी चल रहा है.
हाँ मैं बंद कर दूँगा. Live streaming end करने पे अपने आप विंडो जैक नहीं होगा?
नहीं. You have to stop it.
अच्छा, how to do that?
3 dots